So here we are, section 5.7, Prokaryotic Structures, where we'll go through all the bits and pieces of the bacteria, or the prokaryotes. It's a bit of a doozy, so I hope you're comfortable. Sit back, enjoy, try not to fall asleep. First off, we know from back in Bio 101 there are two classifications of cells: prokaryotic and eukaryotic. Now, those prokaryotic cells are like your bacteria and archaeans. Those are the ones we're going to be talking about. They have no real compartmentalization. Now, sometimes sections of their of the cells are invaginated to create little pockets, but they don't have real membrane-bound organelles. We've learned they have relatively simple structures in the past, but as we'll see today, those relatively simple structures can get kind of complex. And then eukaryotes are just about everything else. They're the ones that have a membrane-bound nucleus where they keep their nucleic acids. They have a lot of compartmentalization, and they have very, very complex structures. All prokaryotes have three structures in common, as in every single prokaryote out there has three things. They've got a plasma membrane, they've got ribosomes, and they've got a nucleoid region where they keep the rough area where they keep their nucleic acids. The plasma membrane, as we learned about before, is a phospholipid bilayer. It's got two layers of phospholipids with a bunch of proteins and carbohydrates embedded within it. It supports transport as well as signaling other cells. The nucleoid region is the area where you're going to have the genetic material of the cell. In bacteria, that genetic material is DNA, just like it is in you. It's not like there's a nucleus, like in your cells, where there's a membrane-bound or double membrane-bound organelle that holds that genetic material. In this case, think of this in a bacteria, it's a circular piece of DNA that's basically hung up on hooks around the cell. And then we have ribosomes. And like you guys know, ribosomes are little protein-making factories. They churn out proteins by reading ribonucleic acid, RNA. So if you recall, in the central dogma of molecular biology, you've got DNA, and that DNA is going to be transcribed into RNA, and the RNA gets translated into a protein at the ribosome. So eukaryotic cells have ribosomes, and so do all prokaryotic cells. Once we get through those structures that all prokaryotes have, we have a few structures that are sort of optional structures. They're bits and pieces that some prokaryotes have and others don't. So in this particular lecture, we're going to talk about pili, flagella, a cell wall, and my personal favorite, and you'll hear it a lot, is glycocalyx. I like glycocalyx. It's not just fun to say. It's got an amazing array of uses. In the future of the class, be on the lookout for any time glycocalyx comes up, because it's not just bacteria. We see it in protists and in animals, not so much in plants, and it does all sorts of cool stuff. But I'll get off that, and we'll talk about glycocalyx many, many, many more times. For now, let's just focus in on the cell shape. We talked about the membrane of the cell. Prokaryotes are all identified by a particular shape. Now, if you recall, the membrane of the or the shape of the cell is produced by the cytoskeleton. And these intermediate filaments link to each other. They sort of press up against each other to create a tent-like network. Prokaryotic cells can be classified as cocci, bacilli, vibrios, spirochetes, or sporili. As I just mentioned, intermediate filaments networked across the cell are going to control the ultimate shape of the bacteria. They're going to give it that round shape, or that long shape, or that spiral shape, or that Cheeto-looking shape. The first shape we're going to talk about are the cocci cells. You've probably heard of this. We've heard of like Staphylococcus or Diplococcus. Think of your sore throat, right? Streptococcus, strep throat, right? You see where it's going here? So cocci are spherical. So they're big balls, and they can be arranged in different ways. If they're singular, it's coccus. Cocci is or cocci is plural. So you have diplococci, um, staphylococci, streptococci, sarcenas, which is totally different, and tetrads. Tetrad meaning four, so you got four of them bumped together. Sarcenas, which are a quad put together, and so on. So we got spheres. We've got bacillus. Bacilli are rod shaped. So they've got they're elongated. They're like little tic tacs. And again, you see the same diplo, meaning two are connected. Strepto is a big long chain, and so on. Vibrios are comma shaped. They are the Cheeto of the bacterial world, but they don't taste like Cheetos. And yeah, they're really not terribly much like Cheetos aside from the shape. Spirochetes are spirals that are flexible. So think about uncooked rotini, hardened, strong. They don't bend very much. Versus spirilli, which are very flexible spirals. They wet noodles. So there you go. The 5 different types of shapes. Fully expect that you're going to need to be able to match the shape to the name. Maybe I'll just give you a picture of a bacteria, and then you've got to figure it out. So if it's Cheeto shaped, it's a Vibrios, and I have no earthly idea why I'm so obsessed with Cheetos right now. It may have something to do with I'm hungry, but yeah. So suffer with me on that one. Cheeto shaped or Vibrios. Now, I said earlier, and I think I said in Bio 101, that prokaryotes aren't very complex. They're simple structures. Well, now that you've grown up a little, you know the world's not always as cut and dry as that. Prokaryotes can be very complex, and they can grow that complexity through what are known as invaginations. Invaginations is a pocketing of the membrane. So those along the edge of the membrane, you have actin filaments that can deform to create a pocket, and then that pocket is reinforced with intermediate filaments. So you can go from just a typical blob-like cell on the left to something a bit more complex that's on the right. That creates sections of the cell that could have chemically distinct properties from other parts of the cell. I think as you learned about last semester, we talked about anaerobic respiration. Anaerobic respiration occurs because you're able to form an electrochemical gradient right in that pocket right there. So invagination allows prokaryotes to be a little bit more complex than we would normally give them credit for. One level of complexity that forms is the thylakoid structure. Prokaryotes can have thylakoid structures, which are sort of folded membranes. It's basically, think of invagination after invagination after invagination to create like a rib cage type effect. It's almost like the cell is squished into little plates, so that there's a lot more surface area that chlorophyll can be sitting in order to trap light. Remember, in biology, whenever you have twists and turns, when you have more of these structures, you're going to have more surface area, and more surface area typically means more biological metabolism. Another type of specialized structure that can form in these little pockets are called magnetosomes. Magnetosomes are pieces of the cell that actually can have an iron crystal inside of them, and that iron will naturally line up to the Earth's magnetic field. It lets some organisms know true north from true south. Obviously, bacteria can use this, but so can birds in order to figure out which way to fly. That crystal inside, growing in these little pockets, will act like a compass. And some bacteria can have these nucleus-like bodies. Basically, through invagination, you can create a double membrane-ish surrounding the nucleoid region. And this is kind of neat. If you look closely at this, you see invagination that creates a double membrane around the nuclear material. Well, we know that the nucleus itself is double membrane bound. And similar to this image, it has pores allowing pores in order to allow some things out of it and some things into it. So this is almost one way that we think there may have been the initial formation of the nucleoid. I'm sorry, the nucleus in the nuclear membrane. Next optional structure is glycocalyx, and you know I love glycocalyx. Glycocalyx is really thick and it's slimy. It's this coating of polysaccharides and proteins and lipids, nucleic acids. Basically, it's all that leftover crap that's in the world. It's heck, think of it almost like phlegm. It is sticky and wet, and it surrounds a lot of bacterial cells. What does it do, me, you might ask? I'll tell you. It can hold cells together in order to exchange DNA. It can exchange chemicals. It can help bacteria float in water. It can repel attacks like a shield. It can bind nutrients to itself, to the bacteria, and it can protect from ultraviolet radiation. Now, if you didn't get all that, don't worry. I'm going to go through them a little bit slower. First off, it can hold cells together. Remember, bacteria can send this bit of DNA back and forth across what's known as a pilus. So they'll literally touch each other with an appendage, and genetic material will flow from one cell to the other, and that takes a little bit of time. So glycocalyx will hold the two cells together while their pili are attached in order to allow that DNA exchange. So it's sticky and holds things together. It helps some bacteria float in water. If you look at the screen, you can see a little kid there swimming. And he's got these water wings. And I don't know if you remember water wings. Water wings were the things you put on, and they made you feel like you had muscles. And that was like the last time in your life you felt like you had muscles. And they help you float. Well, what do you fill those with? You fill them with oxygen. Well, not oxygen. You fill them with carbon dioxide. You fill them with air. That makes them less buoyant than the water around them, or I'm sorry, more buoyant than the water, the water around it, and they float upwards. Bacteria likewise can pump gas into their glycocalyx, and it's almost like blowing a snot bubble. It fills up this glycocalyx with a gas. That glycocalyx expands, and it causes the bacteria to float. And this means the bacteria don't need to use energy to swim; they can just float. That's very useful, as you can imagine, for cyanobacteria, or bacteria that need light for photosynthesis. Gets them closer to the surface where they get more light without using energy. Glycocalyx can act like a shield. Basically, think about bacteria that you eat. They're going to drop into your stomach, and what's in your stomach? In your stomach, you got gas, right? You've got acid. That acid will begin to break down the glycocalyx over time. For some organisms, that's the end. There's very little glycocalyx, so the bacteria gets digested very readily. For others, though, they have a very thick layer of glycocalyx that allows them to survive in the stomach until they can get passed on to the next level. It's sort of like that shield gets lower and weaker and weaker and weaker, but oh, you made it into the intestine, and now you can infect the person, and then they puke all the time. You can use glycocalyx to bind nutrients. Obviously, I told you it was sticky like phlegm. If you think of it like you're chewing gum and the gum pops out of your mouth. I don't know if you've been there, but I know I've at some points been chewing gum, and then magically the gum like basically falls out of my mouth. It's not like I spit it out. It just sort of, I don't know, it has a mind of its own, jumps out of my mouth, lands on the ground. Well, when that happens, do you pick up the gum and put it back in your mouth? I have no idea who I'm talking to here, so I don't know, you might. But most people don't pick gum off the ground and put it in their mouths. If you're listening to this and you are the person that picks gum off the ground and puts it back in your mouth, don't do that. That's that, you know what? Never mind. You be you. You be you. You put that back in your mouth and do whatever you want. But the reason most people, okay, back to the point of this, the reason most people don't put gum in their mouth after it's fallen on the floor is because it's covered in gunk now. If I'm at my house, I don't know, it's a cat running around or something, it'll have hair all over it. It'll have disgusting material. I don't keep my floor terribly clean. I can't help it. Well, I guess I could help it if I clean more, but that requires energy and not talking to you guys. But there's gunk all over it. We don't put that right back into our mouth, but bacteria, they can, as they're going through the environment and picking up this debris, they can then, over time, start digesting that debris, pulling it through that glycocalyx into the cell to digest, to get nutrients out of. So it's almost like throwing a net out into the environment and picking up some nutrients, some snacks while you go. And the bacteria act to serve as ultraviolet radiation. Now remember, ultraviolet radiation causes mutations. It's why tanning is not necessarily the best idea, because literally you're creating photodimers that will lead to skin cancer. And if that skin cancer is caused by changes in the DNA. Bacteria don't necessarily want changes that could be harmful. So they don't want to change their DNA too much, as a bacteria could want anything. So instead, they have this glycocalyx on the outside that acts like sunscreen, preventing solar radiation from damaging the DNA. So those are all the different optional bits and pieces, and especially that is what glycocalyx is useful for. Sort of an undersung function of glycocalyx is the creation of biofilms. Biofilms occur when glycocalyx stick bacteria not just to surfaces, but to each other. They create these favorable surfaces, favorable environments for the bacteria to grow, these very localized environments. Here on the screen, you can see basically a bacteria will bind to a substrate. It'll hold on using glycocalyx. Then it'll start to grow. The glycocalyx prevents it from washing away. If you've ever been in a pond, in a stream, you'll notice that some of those periphytic algae that are sitting there, which we haven't covered yet, we will soon, are they're these green things that are growing on rocks, and they don't wash away with the current. That's because they've got glycocalyx holding them in place. Over time, the population builds up large enough until they can have some sort of group response. It's called quorum sensing, a group response that will be triggered by the amount of nutrients in the cluster of glycocalyx. With quorum sensing, each and every one of those little bacteria are beginning to secrete some small molecule. Over time, the more organisms that are then, the more of those molecules build up. When the molecules build up to a certain level, it enables a group response. You've probably experienced quorum sensing when you've gotten sick. I don't know about you guys, but sometimes when I get sick, I wake up in the morning and I'm like, oh, I'm going to feel sick later. And then it's just a really rapid drop-off. Well, you were sick, likely. You had this bacteria in you for quite some time. It wasn't just that suddenly this happened. It was just that suddenly the bacteria signaled to each other bacteria to switch their life phase, to turn on to a different, basically to switch functions, go from being not very dangerous to, oh God, I'm sick now. Probably the most common example of a biofilm you have experienced is dental plaque. I hope you all brush your teeth. If not, my PSA for the day is brush your teeth, because dental plaque is a type of microorganism that secretes glycocalyx and holds onto your teeth. It takes roughly 12 to 24 hours for that glycocalyx to solidify into an almost concrete-like structure. You've got 12 to 24 hours to mechanically remove the glycocalyx before it becomes so hard that it can only be scraped away. And if you've ever been to the dentist, which I hope you have, you know that they take that little instrument and they scrape some stuff away. What they're scraping away is the solidified biofilm that is built up on your teeth. So brush your teeth at least twice a day. Try and floss, get that plaque out from the center. Because if you don't do that, it will continue to build up until you have a serious medical condition. So we've talked a bit about glycocalyx, and let's talk about the cell wall, which is oftentimes right underneath that layer of glycocalyx. Most bacteria have a very rigid cell wall right outside of their plasma membrane. So it's not just plants. You guys remember plants. Plants have a cell wall. You learned about that in 101. and that gives them that square shape. Now, bacteria don't typically have square cell walls. As we saw, there are many, many different types of bacteria. But bacteria use their cell wall for the same thing that plants use their cell wall for, which is to avoid changes in osmotic pressure. So if you put the bacteria into a hypotonic solution like pure water, the water won't just flood into the cell and make it burst like a balloon. Or if you put it into salt water, it's not going to just shrivel up and die because you've got that cell wall to protect it, especially in pure water conditions, hypotonic conditions. Most cell walls in bacteria are made up of peptidoglycan. Peptidoglycan is a bunch of carbohydrates that are cross-linked. The way I always think about this is, I don't know if you guys remember ever building like a fort or something out of popsicle sticks, but basically you've got a few ways to build a fort out of popsicle sticks. One way to build a wall out of popsicle sticks is to take a dab of glue and put the popsicle sticks edge to edge and just make one solid sheet. And I don't know if you've done that before, but it's not terribly sturdy. If you take it and you can, move it around, it's going to break. It's going to break very, very easily. How do you strengthen that wall that you've created with popsicle sticks that are glued side by side by side? Well, the way you strengthen it is you put a popsicle stick perpendicular to the other ones, sort of at an angle so that it goes across all the others. Put glue on the back and put it together. I'm basically teaching you, here's how you build a popsicle stick fort. But basically, you create a fence. Heck, think about fences in your neighborhood if you happen to have any, or as you drive around. What you'll see is not just planks stuck in the ground next to each other, stapled onto each other, but you'll see planks, vertical planks, and then there are horizontal planks connecting them all. That's what peptidoglycan is. It's those vertical linkages with amino acid cross links. So up and down peptidoglycan, and which is so think carbohydrates that are up and down, and amino acid or polypeptides going across to link them together. It's in this outer wall, this cell wall, that we can really get to our first major classification of bacterial types. You got gram-positive and you've got gram-negative. Now, most people, when they've heard of bacteria, they've heard of gram-positive and gram-negative. These two types are distinguished by their type of cell wall, how thick their cell wall is. And it's distinguished by what's known as gram staining. So you got those two types. Type 1 is gram-positive. Gram-positive has the cell membrane and then a thick cell wall of peptidoglycan. So gram-positive, this sounds stupid, but here's what I always think of. I always think of pumpkin pie. Pumpkin pie is basically, it goes crust and then pie filling. That pie filling, think of as the peptidoglycan. So you have a cell membrane on the bottom, the crust, and then pie filling, the peptidoglycan. It's exposed. There's no additional layer on top. So it's gram-positive. That gram stain will actually bind to the peptidoglycan. Gram-negative are kind of neat. I think of those like apple pie. You're going to have a layer of a membrane, then you're going to have peptidoglycan, and then another membrane on top, like an Oreo-type squish thing, or in this case, an apple pie. You got crust on the bottom, this nice filling, which would be the peptidoglycans, and then a crust right across the top. This stain's negative because the gram stain cannot get to the peptidoglycan. It hits that crust at the top, the other membrane layer, and doesn't stain. So gram-positive has a thick peptidoglycan layer, like pumpkin pie. See, they all start with P. Gram-negative is going to have a peptidoglycan layer sandwiched in between two phospholipid bilayers. So gram stain will end up allowing us to see what type of cell wall these have. And that has a big impact on antibiotic resistance. Gram-positive bacteria are usually very susceptible to antibiotics. It makes sense because an antibiotic will hit that peptidoglycan and start to diffuse across the membrane. Otherwise, there's membrane, peptidoglycan membrane, and the that antibiotic or that metabolite of an antibiotic doesn't get all the way through. So when you think gram-positive bacteria, those are susceptible to bacteria. I'm sorry, to antibiotics. This is why your doctor typically wants to know what kind of bacteria you have before prescribing you an antibiotic. If the antibiotic that if the infection you have is a gram negative, that is, antibiotics won't work well on it, the doctor doesn't want to describe an antibiotic because that antibiotic might cause more damage than it helps. And then we get to how do bacteria move around? These are the last two optional structures we'll discuss. They can use flagella or they can use pili. Now, flagella and pili both have very, very similar structures and functions. A flagella, as you probably learned about in Bio 101, is this whip-like structure, but it operates on a totally different basis. What ends up happening is it has this. I wish I could describe it. You're looking at it on the screen, but you have to actually see it move. You've got a rotor, some sort of rotationary device, group of proteins embedded in the membrane. And that actually is free spinning. Attached on that yellow piece there, you'll have motor proteins. So those motor proteins will just grab on and pull. And then they let go and they grab and they pull. And it begins to get this thing moving. It's almost like when you were younger, you know that it's like a wheel of death in the playground. You'd get on it and you sit like in the middle, and then your friends would start to grab the edges and pull and throw and pull and throw and pull and throw until you got this thing moving fast enough that everybody flew off and died. And it was usually rusty. I'm not sure if you know the one I'm talking about. It operates on the same principle. Somebody on the outside grabs and throws, grab and throw to get this thing spinning. On the top, you have a long filament that's basically got an intermediate filament inside of it and a hook. So it's got an L turn and then a big long whip. As it spins, that whip moves in a circle, and that creates a motor force, just like a boat engine would. So think of a speedboat where you can get this thing moving very fast for a small bacteria. That can really get that bacteria moving forward. The motor proteins can move it in reverse in order to pull it backwards. And then finally, we have pili. Bacteria can use pili for two things. We talked about one earlier, which is transferring genetic material if two pili touch each other. The other is basically to grab a hold of a substrate and pull yourself forward. Grab a hold of a substrate and pull yourself forward. Think of it as, I don't know, like you would picture an octopus crawling on land. They reach out, they grab, they pull. They reach out, they grab, and pull. Pili are going to be assembled or disassembled in front of or behind the cell in order to move it by pushing or pulling. Pili, like we said, also can also be used to transfer genetic material. So with flagella, you can move the bacteria forward and backwards. With pili, you can pull the bacteria forward or push it backwards.